Vision perception, a comparison between how protostomes and deuterostomes perceive vision. Presented by Janae Hall, Dennis Strickland, Kirsten Tompkins, and Paige Tarbett. When we break down the concept of vision, we start with the most necessary subject, the eye. The eye is a structure that has the ability to receive and distinguish light while outlining spatial detail. Beginning its evolution over 700 million years ago, animal vision started with the simple task of detecting light in order to determine day from night. As animal vision began to evolve due to changes in their surrounding environment, it was done so in small steps but provided advantages over other species in the race for survival. Evolution likely evolved from simple to complex. An experiment conducted at Lund University started with a flat, light-sensitive patch, gradually adding over 1,800 modifications such as forming a cup, adding a lens, and constricting the opening. This concluded that these eye improvements could have taken place over the span of about 360,000 generations. Eyes show a wide range of adaptations to meet the requirements of the organisms that bear them. Eyes vary in their visual acuity, the range of wavelengths they can detect, their sensitivity in low light levels, their ability to detect motion, and whether they can distinguish color. Before we begin to compare protostomes versus deuterostomes, first we need to briefly understand the main parts of the eye. First, there is the pupil, the hole at the center of the iris which allows light into the eye. Human eyes have circular pupils, which are actually fairly ineffective at blocking light. With slit-shaped pupils, animals can exclude light more effectively. This is generally found in animals with a nocturnal lifestyle that need to protect their eyes during daylight. Shown also are animals with W-shaped pupils, as well as rectangular and crescent-shaped pupils. Next, we get to the lens. When referring to eyes, terms such as simple eyes and complex or compound eyes refer to the number of lenses within the eye itself. In simple eyes, there is a single lens. This is assumed to be the first eye format. In complex or compound eyes, there are multiple lenses, each focusing the light that they receive on a small number of retinal cells at the back of the eye. Next, we cover the retina, which works much like the film in an old-time camera. Each species of animal have a retina adapted to the light conditions dependent on whether the animal lives in a diurnal or nocturnal habitat. The retina is responsible for receiving and processing visual information. Finally, we cover the optic nerve or optic ganglion. This structure serves as a railway system for visual information to quickly and directly be transmitted from the retina to the center of the brain or the visual cortex. We will now discuss the different kinds of vision found in both protostomes and deuterostomes. We will begin by discussing vision perception in protostomes. We must first define what a protostome is. By definition, a protostome is any major group of animals defined by their embryonic development in which their first opening within the embryo becomes the mouth. At this stage of development, the later specialization of any given embryonic cell has already been determined. Protostomes are one of two groups of animals that have a true body cavity. Protostomes include mollusks, annelids, and arthropods. We will discuss two different types of vision found in protostomes, beginning with the compound eye found in arthropods. Typically, arthropods have a pair of compound eyes that are composed of closely packed facets called ommatidia. Ommatidia are the structural and functional units of vision in the compound eyes of an arthropod. The number of ommatidia varies considerably from species to species. Externally, each ommatidium has a lens that is formed by a convex thickening of a transparent cuticle. Beneath the lens, there is a crystalline cone that functions by refracting incoming light down into a receptor region that contains light-sensitive pigments. These pigments function to absorb wavelengths of light and generate nerve impulses through the photochemical process similar to that of vertebrates. Unlike vertebrates, insects cannot form a true or focused image of the environment, so their vision is relatively poor. However, their ability to sense movement by tracking objects from ommatidium to ommatidium is superior compared to most other animals. Many insects can distinguish 200 images per second, while in humans, still images blur into constant motion at about 30 images per second. Next, we will discuss the camera eyes found in some mollusks. There are 4 classes of mollusks that possess eyes that vary in size and structure. Their eyes range from simple pit eyes to compound eyes, pinhole eyes, reflector or mirror eyes, and camera eyes. We will discuss the cephalopod camera eye, which strongly resembles the camera eye of vertebrates, mostly in fish. The remarkable similarities between the camera eyes of cephalopods and vertebrates is one of the best-known examples of evolutionary convergence. The cephalopod camera eye consists of 2 chambers that are separated by a lens that functions to project an image onto the light-sensitive retina. The retina in the cephalopod camera eye closely resembles that of the vertebrate retina, although it is not as sophisticated. In both vertebrates and cephalopods, the eyeball is located in a socket and is rotated by extraocular muscles. Both vertebrates and cephalopods have a pupil equipped with an iris that controls the amount of light that passes through the lens. In fish and cephalopods, the lenses are hard and circular and must be transparent. For this purpose, crystalline proteins have been independently recruited. The class of crystalline is different in cephalopods compared to that of vertebrates. It is believed that their structural properties are necessary for the lens to function properly. The lens functions to focus light. In a high-powered spherical lens, there is risk of spherical aberration resulting in blurred images on the retina. Both cephalopods and vertebrates have come up with the same solution to this problem. The refractive index of their lenses gradually changes from periphery to center due to different concentrations of crystalline, and therefore the lenses achieve high sensitivity and exceptional imaging. Now we will discuss how deuterostomes perceive vision. By definition, a deuterostome is defined by their embryonic development, in which the first opening of the embryo becomes the anus. At this stage in their development, the later specialized function of any given embryonic cell has not yet been determined. Deuterostomes, much like protostomes, have a true body cavity and include animals such as echinoderms, hemichordates, and chordates. Snakes can see heat signatures at night. Snakes are very nearsighted and typically have trouble seeing things that aren't moving. That's where other senses such as smell and heat signature come into play. Pit vipers, pythons, and some boas have infrared-sensitive receptors in deep grooves of their snouts, which allow them to see the radiated heat of warm-blooded prey mammals. Rattlesnakes can see in infrared at night and also detect their prey's body heat. Chameleons' eyes are shaped rather unusually. The chameleon does not have an upper or lower eyelid. Instead, they have one large eyelid that is shaped like a cone. Because of this cone shape, their eyes move as though they were mounted on turrets. Each eye moves independently from one another, allowing the chameleon to see what is in front of and behind them at the same time. Fish eyes are lidless, and the lens is round and close to the cornea. Their retinas generally have both rods and cones, which means that most species of fish have colored vision. Some fish can see ultraviolet light, while a few other species can see polarized light. The lens within a fish's eye is considerably thicker, almost spherical, to increase refraction. Focusing requires moving the lens forward or backward, much like you would use a magnifying glass to focus in and out. The eye of a bird closely resembles that of a reptilian's eye. The eye is not spherical, but rather is flat, which enables more of the bird's visual field to be in focus. Birds see ultraviolet light. Ultraviolet vision can be useful in their courtship. Many birds show plumage patterns in ultraviolet that are actually invisible to the human eye. This means that some birds whose sexes appear to look similar to humans may be distinguished by the presence of ultraviolet reflective patches on the feathers. Rat vision is quite blurry. Normal pigmented rats show vision measuring around 2600. Albino rats are either blind or severely visually impaired, with vision measuring around 1200. Rats, much like birds, and those few fishes mentioned previously, can see ultraviolet light. However, ultraviolet vision in rats is presently not well understood, considering most rodents are nocturnal. This unusual and unexplainable finding is currently an active area of research. Horses have the largest eye of any land mammal. The visual acuity of a horse is about 20/30. Horses have dichromatic vision. This means that they can see blue and green colors of the spectrum, but they can't see red. Many domestic horses are nearsighted, while wild horses are usually farsighted. Because of the location of the horse's eye on the side of the head, they generally have a 350-degree range of vision. This, partnered with their large eye size, means that they are very sensitive to the slightest of motion. This shows to be advantageous in that the horse can first detect potential predators in their periphery or from nearly behind them. However, the vision in this periphery is not very clear, which presents itself to be a problem on windy days. If there is too much movement in their peripheral vision, but not enough clarity, it is difficult to distinguish between a blowing tree limb and a stalking animal. Finally, we have a slide that just lists a few fun facts that we found along the way as we researched our topic. Hope you enjoyed, and thank you for watching.